Okay, let me move on to reading your questions in the chat. One second here. All right. Okay. Okay, I'll just read these. For short read library prep, is there a particular method you recommend? For example, Nextera, Nextera XD. Okay, so sorry I didn't make this clear, but the short read prep part of our library, the part that turns the long read, the barcoded long reads into short reads, is actually included in our prep. So you don't need any other reagents like Nextera, Nextera XD, or any other short read prep enzymes or system besides our kit. So our kit already includes all of that, includes everything you need in order to transform your short reads into long reads and to transform your long molecules into the libraries that I mentioned before. So you really don't need to get anything else or worry about whether Nextera is better or any other sequencing method. It's all provided in the kit. I hope I answered that question. Let me know if you meant something else. What if your sample isn't homogeneous? Not sure what you mean here by not homogeneous. Maybe if you could clarify that, and I'll get back to that question later on. Are the barcodes integrated at specific intervals from the priming site or at random distances? That's a very good question. So the barcodes are integrated randomly, but the distances, the average distance between every two barcodes is actually not random. So we can control the length or the intervals between the insertion points, but the actual insertion points of the barcodes are completely random. And there's no sequence specificity or no sequence preferences. It's completely random, and we can control the intervals between them. Moving on to the next question. Can you share an example informatics report of the data produced from known and unknown samples? Yeah, definitely. So first of all, I can show some of that now. And also, if you go onto our website, and I can share those links later, there is a place where you can download an example report of both our long-read FASTQ files and of a CSV file that summarizes the downstream analysis of the long-read FASTQ file. And that includes classification information, quantification information. So that can be downloaded from our website, but I'll also run you through that report in a few minutes, at least parts of it, because running through the entire report will just take too much time now. Yeah, another good question here is, what about library quantification normalization prior to sequencing? So that's a very good question, and we've dealt with that in several different ways. So first of all, all the input into every sample needs to be 10 nanograms. So your libraries need to be quantified using qubit to 10 nanograms. So 10 nanograms is the input genomic DNA into each barcoding reaction. So that's one level of normalization that we do between different samples. But 10 nanograms can still mean different things for different samples because there may be some contaminant host DNA. So it doesn't, the fact that you're measuring 10 nanograms on qubit doesn't mean that you actually have 10 nanograms of barcodable DNA. And that could potentially lead to different numbers of unique molecules being sequenced from each one of your samples. So if you have 24 samples and they're all normalized to 10 nanograms, you might have some variation in the number of molecules that were sequenced between those samples. What we do include in each and every kit is a standard reaction for qPCR, which you could use in order to normalize the number of molecules that you sequence from each sample to be whatever you want. If you want them to be equal, they can be equal. If you want them to be, you can normalize them to be whatever you want. So that standard, which essentially measures the number of barcodes that you have in each and every sample, is included in the kit. But what we recommend, at least as a first try, is to simply normalize all of your samples to 10 nanograms. And if you're not happy with that normalization based on the data, in your next run, you can go through the slightly longer route of qPCRing each barcoded sample separately and normalizing it separately. I hope that answers your question. But generally speaking, I think that especially for high biomass samples like stool or soil, where most of the DNA that you're measuring is actually barcodable microbial DNA, the variation from sample to sample, assuming you're starting from 10 nanograms, should be relatively low. Okay. Moving on to the next question. How many reads per sample do you recommend, let's say, for a human gut metagenomic sample? Yeah, that's a great question, and there's no one answer to it because different people have different applications. Even for a gut microbiome, there may be variation in how deeply people want to look into a sample. But just based on what our customers in the gut microbiome space are looking at, they generally want to look at anywhere between 5 and 30,000 unique long molecules. Now, I realize that that's hard for people that haven't been doing single molecule sequencing so far. It's hard for them to understand what the equivalent for that in their regular Illumina short read sequencing. Because, say, for example, now you're doing 1 million reads per sample. How many actual unique single molecules is that? It's hard to tell. You don't really know if that's 1,000 unique molecules or 100 unique molecules. How many PCR duplicates do you have in your data? The difference with our data is that you actually know how many unique molecules you have. So what we're seeing people in the gut microbiome space do is sequence anywhere between 5,000 and 30,000 unique molecules. But obviously that's completely up to you. And you can tune the complexity of how many unique long DNA molecules you're sequencing in our protocol. So if you go through our manual, there's a place in the protocol where you can decide how many barcodes or how many unique molecules you're going to sequence from each one of your samples. Basically, that decision is made when you're deciding how much volume you're going to carry forward from each one of the barcoding reactions into the pool of all the 24 or 96 samples. Okay, moving on to the next question. What is the input DNA amount needed for the microbiome prep kits? Yeah, so I mentioned that. That's 10 nanograms per sample. That's the input. Next question, does the DNA need to be sheared before ligation of that adapter, or does the adapter include the cutting site that specifically recognizes the start of the 16S gene? Yeah, that's a great question. So the answer to that is that you don't need to do anything. You don't need to shear or anything. The input into the kit is simply extracted microbial genomic DNA. The kit includes everything that you need in order to target the 16S gene. There's no shearing required on your end. Moving on to the next question. All right. Can you give us an idea of how much species diversity you are seeing versus a conventional 18 or 28 ribosomal populations, and how much more complexity will there be on the metagenomics, and how deep will the sequencing need to be? I think that's kind of it's a very good question, but I think the answer for that is will be best answered with our future webinar on shotgun metagenomics and the cons and pros for that versus long read 16S sequencing. So I'd say kind of the general answer to that would be that we think that there's room for both long-read 16S sequencing and long-read metagenomic sequencing. They're simply used for different purposes. Obviously, long-read 16S sequencing is more economic or more cost-effective in terms of sequencing cost. But with the shotgun metagenomics, you can access data that will be hard to access using 16S, even with 16S long-read sequencing. So let me kind of defer the answer to that question to our shotgun metagenomics sequencing webinar. Is your kit formatted for a single run only, or can you split the 24 samples into multiple uses of the kit? Great question. The answer is the kit is formatted for a single run only. So it's a single shot 24 sample kit. Obviously, you can use it for less samples if you want. You can process, 18 or 16 samples, but the kit is a single use. So you can do up to 24 samples, but obviously you can do any number up to 24, but you cannot use it twice. We do have plans in the future for making versions of this kit for less samples, for example, for 8 or for 16. But in the meantime, if you only have a handful of samples that you want to process and you don't want to get a 24-plex kit, you can simply send those samples to us for sequencing as a service, and we'll be happy to process those for you, any number of samples, even a single sample. Yeah. So there's a few comments here that 10 nanograms is fine for high biomass applications like soil or stool, but for other types of samples like low biomass samples, like for example skin, 10 nanograms is way too much. And I completely agree with you. This is our high biomass kit, and we have a low biomass kit in the works, and we think that will be. We estimate that will be out within, I'd say, 6 to 8 weeks. That kit will accept input amounts that are lower than 1 nanogram, so as low as a few hundred picograms per sample, and that will be specifically designed for samples like skin samples or other types of biopsies that have higher human contamination and lower actual biomass. And the primers that we're using for the targeting here actually have a 100-fold targeting bias for microbial 16S versus human mitochondria. So if you have a sample, let's say a skin microbiome that has, let's say 50% human DNA and 50% microbial DNA, in your actual 16S data, you'll see the microbial 16S 99-fold enriched compared to the human mitochondrial 16S. So, so that's just to address the comments that 10 nanograms of input would not be very useful for low biomass samples. That kit is not out yet, but we are already providing that as a service. So if you have skin samples or any type of low biomass samples, we are ready to process that as a service, and as a kit that will be ready within a few weeks. Yeah, how do you distribute the barcode randomly within the molecule? It's a great question, and that's the part of our chemistry that was developed for almost a decade in multiple research institutes. A lot of that technology is proprietary, but what I can say is that we've developed a set of enzyme, or I'd say collectively an enzymatic system that enables to take a barcode sequence and then copy and paste it randomly into the same molecule intramolecularly. So the system is not transposon based at all, but I find that that's kind of a good analogy for people to understand. You can think of a transposon system as a system that takes a sequence and that inserts it randomly into DNA. Our system does the same, only it does it intramolecularly. So it copies and pastes a unique sequence into different positions within a molecule, but it does it intramolecularly as opposed to intermolecularly like a transposon system does. but our system is not transposome based. It's based on other enzymes or enzymatic system that we've developed, and that's how we're distributing it intramolecularly. Great. So I think I've went over all the questions that were on the chat section, and this would be, I think, a good opportunity to maybe run you through some of the data analysis and how that looks like, and also open the stage for your questions. Okay, let me just see how I can share my screen so you can see the data. Maybe this could be, you know, so maybe you guys can ask questions now, and in the meantime, I'll prepare some of the data files to show you. So you can unmute yourself if you have a question right now, and go ahead and ask any questions. Great. Okay. So if we answered all the questions that you had, that's great. Let me just run you through. There were a few questions just relating to what the actual output looks like. So I won't go over the entire output files because they include a lot of files, a lot of visualizations. Let me just go through some of the most important or fundamental data files. So you can see the FASTQ file right now, right? Yeah. So this is the output from our pipeline. This is the raw FASTQ long read file. So each header here would have a serial number for that molecule, which would be unique, and then followed by the actual sequence of that molecule. So you can see by the length here, this is a full-length 16S molecule, about 1,500 base pairs. And the format here is the regular FASTQ file format, so followed by the quality scores for that molecule. We're generating a FASTQ file so that you know, it's the industry standard, so that you can use this file as input into your existing pipelines and run your own analyses on them. So this is we generate for each one. So for example, in a 24-plex in a kit, you get a folder, a zipped file with 24 folders in it, one for each sample. Within each one of those folders, you will get a raw long read data file like this. A FASTQ file will also provide a FASTA version of this file. And for each one of the samples, we'll also include a CSV file like the one that you can see here. This is just an example of those. Let me run you through what you're seeing here. So this is a CSV file describing the downstream analysis of the long reads from one particular sample. Every row in this sample represents a single molecule from your original sample. That means a single sequence from your FASTQ file. So for example, if you're interested in molecule number 1533, for example, you can go to your FASTQ file, search for molecule number 1533, there it is, and that is its sequence. And if you're interested in additional information about that molecule, you can see here what that molecule's ref ID is, what the reference length is, what is the length of the molecule that we were able to reconstruct using our long read reconstruction. And then you have different statistics about that specific molecule. For example, what fraction of the reference was covered by our long read, the full taxonomy for that specific molecule, and then specific columns for genus level resolution and species level resolution. Whoops, let me do this. And then what the percent ID was to the reference chosen in the database. So this is just an example of the statistics that we generate for each and every single molecule that we sequence. So the sequence, the length, the full identity, statistics about how well it aligns to the reference, to the best reference chosen for it in the databases. Calculating relative abundance is trivial from these CSV files because in contrast to regular Illumina sequencing, where a read just represents a read and not the actual abundance because it doesn't count for PCR biases, in this file, each row here actually represents not a read, but a single molecule, a barcode from your original sample. So calculating relative abundance of species from this file is actually very easy, just using simple, even Excel-based analysis. But we do go one step further and do these calculations for you. And the output folder would include OTU tables and different pie chart visualizations of the classification and the quantification information. Okay, so that was just a summary of what a partial summary of what our output looks like. If any of you are interested in taking a look at what a full output folder would look like with all the different data outputs, shoot us an email and we can send you example data for that. As I mentioned before, you can download from our website example FASTQ files and CSV files like this for the synthetic communities from Zymer Research and ATCC. And if you're interested in the other downstream analyses that we're doing, such as the different visualizations that we're providing, shoot us an email and we'll send you example data. Are there any additional questions on the chat, or that's it? Okay.
Guys, one question, if you don't mind.
Yes, please go ahead.
First off, excellent presentation, both in the way
Thank
it was you sent the data across, but also in terms of the work that you're actually literally could be a game changer in the microbiome field. So congratulations there.
Thank you.
I'm just wondering, can you give an indication as to the price per sample, particularly for the low biomass kit that you mentioned earlier?
Yeah, definitely. So our kit is currently, you can go on the website, you have pricing there. I'll save you the suspense. It's priced at $999 per 24 samples. So that adds up to about $40 per sample. So that's the pricing for our 16S and or our 16S plus 18S kit. And we foresee our low biomass kit to be priced very similarly, if not identically. So $40 per sample.
And if we were to send you extracted DNA to perform the entire analysis?
Yeah, so for the service-based option, it really depends on several parameters, as you can imagine. For example, how many samples you want to process, and even more importantly, how deeply you want them sequenced, because that will affect the sequencing cost. So obviously, the service option would be more expensive, but I can provide you with specific quotes just if you will be able to then offline share that information from me with me. For example, number of samples, depth that you want sequenced, and then I can share a quote with you. But I can tell you that at high volume and relatively reasonable coverage, there isn't a huge difference in the pricing. What we also do offer, so feel free to reach out to us then offline, and we can figure out those parameters for your sequencing, and we can send you a specific quote. What I should also mention about our service is that we have two options for turnaround time. We have a fast turnaround time, which is seven business days, which I think is very fast compared to existing services on the market today. So all the sequencing benefits that we just presented with our long-read single molecule sequencing, you can get them as a service within 7 business days. And we also have a slower turnaround time, which I wouldn't call it slower, I'd call it standard, kind of the industry standard of 30 to 35 business days. And obviously the 7 business day turnaround time is more expensive, but that would simply give you a better selection of options for different projects if they're at various degrees of urgency. So you can choose between 7 and 35 days turnaround time for the service.
That's excellent. Thanks very much for the information. Just one final comment from myself. It would be really, really interesting when you do the shotgun metagenomics analysis so a webinar in the next couple of weeks if we were able to see a comparison of some matched samples via shotgun metagenomics and also via your 16S library prep. The correlation between the two would be very interesting.
Yeah, we'll definitely do that. For all the methods that we're developing, we're always using the same synthetic communities. So the 20 strain staggered ADCC mix and the 12 strain Zymo research synthetic community, as well as complex samples. So we're doing the shotgun metagenomics and the 16S with the same sample, so we'll be able to show comparative data and to show exactly what type of extra information you can get from the shotgun that you can't get from 16S, and in which cases it makes sense to use one versus the other.
Excellent, brilliant. Thank you very much, guys.
Yeah, thank you for your questions. Are there any other questions anyone would like to ask? Okay, so I'd like to thank everyone for tuning in. We're very excited about this launching in this product, both as a kit and as a service. Feel free to reach out to us with any questions about both the kit and the service. We're happy to dedicate a Loop scientist to help you get started with our kit. We do a lot of, I'd say, hand-holding with our new customers, making sure that their first experiment is a success. So feel free to reach out to us and get started with either the service or the kit. And we're looking forward to meeting you and at the different conferences that we go to, and also in our next webinar in a few weeks about shotgun metagenomics and microbial genome sequencing. So thanks again for joining us today, and we'll see you in a few weeks. Have a great day.